Hi everyone, today I'm presenting electromyography data processing impacts muscle synergies during gait for unimpaired children and children with cerebral palsy. I'll cover why muscle synergies matter, what EMG processing choices the authors tested, key results, and why it is important for clinical gait analysis. First, a literature evaluation. Some strengths of this study include using a large sample of patients. This allowed the authors to draw clear conclusions between the relationships of processing methods and the number of synergies. They also took their results to propose a more robust metric, walk DMC. Some limitations of this study is that only 2 pre-processing parameters were tested, ignoring the effect of several other filtering and normalization processes. Additionally, only using EMG data from 5 muscles may limit generalizability of synergy structure, and there is uncertain generalization of these results to other populations such as adults or those with other neurological impairments like stroke victims. This study highlights the need for standardized EMG preprocessing and shows a path via z-score measures like walk DMC for cross-center comparability of results. The impact of this study is demonstrated through its high number of citations, about 98, and its publication in a high impact factor journal. So, why study muscle synergies? Synergies are groups of muscles activated together to simplify controls of tasks like walking in patients with neurological impairments, such as those with cerebral palsy. Fewer synergies are often observed, reflecting reduced neuromuscular complexity. Measuring synergies reliably can improve our understanding of movement impairment and inform interventions. Why analyze EMG processing methods? To extract synergies, EMG must be filtered and normalized. But there is no standard procedure for doing this. Different methods can change the number of synergies and muscle weightings calculated for the same patient. This paper focuses on how low-pass filter cutoff frequency and amplitude scaling affect the number of synergies calculated and the weighing of muscles within a given synergy. So we know what's robust and what's sensitive to change in the processing method. The dataset includes 113 children with diplectic cerebral palsy and 73 typically developing children. The authors did not collect the EMG data themselves, rather they used the data of patients who had gait analysis done at the Collette Children's Specialty Healthcare. For the gait analysis, 5 muscles were recorded per participant. This included the rectus femoris, medial hamstrings, lateral hamstrings, the medial gastrocnemius, and the tibialis anterior. The patients were then categorized by GMFCS 1 through 3, which are listed on the slide. The first processing parameter the authors analyzed was low-pass filter cutoff frequency. The low-pass filter removes the high-frequency noise from the EMG signals. The authors looked at cutoff frequencies from 4 to 40 Hz. The second processing parameter the authors analyzed was the amplitude scaling method. They looked at the difference between normalizing the EMG data to peak amplitude or by normalizing the data to unit variance. Finally, the authors looked at the criteria for identifying synergies, including using TVAF and walk DMC. TVAF stands for Total Variance Accounted For, and walk DMC stands for Dynamic Motor Control Index during walking. Here we see EMG envelopes for 5 muscles over the gait cycle processed with different LP cutoffs. Low cutoffs, about 4 Hz, yield smoother, broader activations. High cutoffs up to 40 Hz retain finer fluctuations. These differences feed directly into synergy extraction, so filter choice definitely matters. These figures from the paper demonstrate that increasing the LP filter cutoff frequency increases the number of synergies identified in both typically developing patients and patients with cerebral palsy. This shows why using a fixed threshold to pick the number of synergies can yield different results depending on the processing method. However, across the range of cutoff filter frequencies, patients with cerebral palsy consistently showed fewer synergies than typically developing patients. This indicates reduced neuromuscular complexity, supporting this theory in literature. Using unit variance scaling was shown to reduce variability in calculated synergy weights across different filtering conditions without affecting metrics like TVAF or walk DMC. This makes unit variance scaling attractive as it helps increase the reproducibility of results across different labs. The implications of the study included reprocessing choices can shift TVAF and the chosen number of synergies. Choosing a hard cutoff for what is considered a synergy can produce different results depending on the processing methods. Walk DMC is more robust to filtering differences, helping cross-clinic comparisons. Standardization of EMG processing is needed if synergies are to guide clinical decisions. And lastly, to address some of the limitations of the paper, for future work, the authors could explore more than just the two processing parameters explored in the paper. For example, high-pass filtering, filter order, and other normalization schemes. They could also include more muscles to see the impact on weighting and groupings, and include different populations to have more generalized results. The main takeaway is that reliable muscle synergy analysis depends on standardized EMG processing. This study shows that results are sensitive to choices like low-pass filter cutoffs, but more stable when using measures such as walk DMC and group-level comparisons. These findings lay the groundwork for protocols that can make synergy analysis both clinically useful and comparable across research centers. This concludes my presentation. Thank you.